My name is Celina Juliano, and I'm an associate professor in the Department of Molecular and Cellular Biology at UC Davis, where I've been teaching, mentoring, and running a research lab for nearly a decade. I've been fascinated by living things for as long as I can remember. I was the kid who brought spiders in jars for show and tell, and I still have a scar on my hand from trying to catch a lizard when I was 12. That curiosity I had for animals when I was young is still what drives me today. I want to understand the incredible diversity of animals and how they can do things that seem almost magical, like regrowing entire body parts, which is specifically what my lab studies. In my research lab, we work with Hydra. It's a small freshwater animal that can regenerate its entire body from just a small tissue fragment, or even from dissociated cells that come back together. What's amazing is that Hydra and humans share a lot of the same genes, so we're trying to figure out, how does Hydra use these genes to do something extraordinary? We hope that by understanding this, we can uncover fundamental principles that one day may help inform regenerative medicine. Incidentally, these Hydra, they also don't get cancer, which is something we are now starting to explore. My lab is primarily supported by a grant from the National Institutes of Health that gives us the freedom to follow discovery-driven science. We're asking fundamental questions like what genes are activated by injury and how do they drive the perfect regeneration of Hydra's body. We also have a second, smaller grant from the NIH to study a closely related species of Hydra that can't regenerate as well and actually does get cancer. So we have two species in the lab, two Hydra species. They look almost identical, but their biology is very different. Comparing them gives us a powerful way to pinpoint the genetic changes behind regeneration and cancer resistance. We have made many interesting discoveries over the last 10 years, but I'll focus on two recent studies that are near publication. In one, we mapped the entire nervous system of Hydra. We know every neuron type, where it is, and we're also learning what they do. This gives us a foundation to understand how the nervous system can regenerate fully from stem cells, which is something Hydra does remarkably well. In another study, we found a way to trigger regeneration in that Hydra species that doesn't regenerate well. That's important because it shows that loss of regenerative abilities can be reawakened. These are breakthroughs in basic science, but they feed directly into our understanding of how tissues and organs might be rebuilt in other animals, including us. Our work is also incredibly cost efficient. About 85% of the funding received by my lab goes to paying the people who do the work, undergraduate students, graduate students, and postdocs who are training to become the next generation of scientists. So the impact of this research is both scientific and human. It's helping build the future of science. Honestly, without this funding, my lab just can't function. My students and postdocs wouldn't be able to finish their research. They might not earn their degrees. Some of them could be forced to leave science entirely. And because so few labs study Hydra, losing our lab would mean losing a hub of expertise and critical infrastructure. We maintain the community resources, publish foundational data, and we drive progress in this field. Without us, that momentum would be lost, and so would the opportunity to unlock Hydra's secrets and apply them to human health.